Hi, everyone. Welcome. Welcome to our next session. I will invite Itai to introduce our speaker. Hey, Itai.
Hey, are we ready to start?
Yeah, you can go ahead with the introductions.
Okay, so hello everyone. I'm happy to have sped through my section to allow Alex to comfortably go through his. Alex is a great postdoc that I have in my lab. He's been doing fantastic work both on the human microcircuit and neuron and synaptic modeling, but also some other projects that maybe in the next conference you'll hear about. But today he will give us a tutorial about the BluePyOp tool that we've been using. It's a nice free tool. I'll just mention that Alex has worked with Dr. Francis Skinner from Toronto Western Hospital, and is currently doing a postdoc with us at the Krembil Center for Neuroinformatics. Alex, the stage is yours.
Awesome. Thank you, Itai. So today I'll be presenting optimizing detailed models of human neurons with BluePyOpt, which is a Python module created by the Blue Brain project. And in this presentation, I'll be giving a lot of details on different tools that I use and repositories that I get data from. So if anyone has any questions about those resources, just send me a quick email and I'll be happy to forward more information about them. So just to give a quick outline of the presentation, I'll first go over a quick introduction to BluePyOpt, as well as some steps that I take in acquiring human data, and then extracting electrophysiological features from that data. And finally, I'll take you through a quick setup that, or examples that I have of setting up an optimization with BluePyOpt. And then finally, I'll just give some quick examples of models that we've developed over the past year. So BluePyOpt, like I said, is a Python module, and it's used to simplify the automation of model optimization. It wraps several different external tools in Python, including the DApp-Python module, which stands for Distributed Evolutionary Algorithms in Python. It includes many different popular evolutionary algorithms, including things like particle swarm optimization, non-dominated genetic algorithms, and indicator-based evolutionary algorithms. It also wraps the neuron simulator, which is a populator simulator used for multi-compartment modeling, as well as the EFEL toolbox, which is another Python toolbox developed by the Blue Brain project. And that's just used to extract electrophysiological features from data. If you want more information about BluePyOpt, you could also check out their paper in Frontiers in Neuroinformatics, as well as their GitHub page, which includes many different tutorial examples on optimizations ranging from point neuron optimizations all the way to multi-compartment modeling. So just to give a quick primer on evolutionary algorithms, now in BluePyOpt, there's two default evolutionary algorithms that are IPEA, which stands for Indicator-Based Evolutionary Algorithms, as well as NSGA2, which stands for Non-Dominated Sorting Genetic Algorithm 2. The basic premise of evolutionary algorithms is to first initialize a population of models with different parameters. Each different color here represents a different model with a different set of parameters. It would then simulate those models and then select from that population of models which ones best capture the experimental data, in this case, the electrophysiology data. And then from there, it would take those best models to generate a new population of models, and then it would cross over some of those parameters between that pool of new models, and then mutate some of the parameters to add diversity to the next generation of models. And so different evolutionary algorithms have different rules when it comes to how it selects different models, as well as generating new models. So the biggest component of this presentation really is acquiring human data, because there is actually a lot of human data out there now coming out with cell type electrophysiology, which gives us some options as to how we want to model that data. Now, one option is to fit the model to a single cell. So with this works for rare cell types where there's no human population data sets available. For that, you really just have the E features from one cell. So you don't really have a mean and standard deviation, which means that with BluePyOpt, which relies on standard deviations to calculate errors, you'd have to set those standard deviations manually, which means you could either set them to very small values to weight particular features in the optimization, or you could take standard deviation values from rodent literature or monkey literature. The quality checks that you want to do is, if you're choosing this option, is to make sure that the cell that you're taking is not an outlier. So that's just making sure that the electrophysiology looks typical of that cell. The E features don't vary too widely from what you'd expect from that cell type. The second option is to actually fit it to population data. If you have access to a large enough population of cells of the same type, you can obtain that data programmatically, and you can, from that data, you can generate means and standard deviation EFIS values as you would normally with BluePyOpt. The quality checks that you want to do with this, though, is to make sure that the protocol is kept consistent. Occasionally, you'll see data sets where the protocol actually changes or it's not consistent across different cells. So you just want to take the same steps of the same amplitude and durations. And you also want to make sure that the quality of the traces are consistently good. Typically, experimenters will discard any data where the cell looks like it's dying or unhealthy, but it helps to still take a look throughout the data to make sure you're not including anything weird in there. So Itai mentioned also that we acquire a lot of our data from Allen Brain Institute. What I do for this is I just go to their cell types database, so celltypes.brainmap.org/data. It should bring you to this search bar. What you would do is just select human, and then you can specify which layer in Cortex you want to take that data from. So here I'm selecting layer 5. Important note about the location. Most human data is taken from middle temporal gyrus, which is resected tissue, usually from people with epilepsy. Occasionally, you'll also see resected tissue from people with tumors, which is not necessarily always in middle temporal gyrus, but for the most part, it's MTG data. You can also play with some demographic data, such as the age, the sex, even the disease. And also, you can specify aspects about the reconstruction. If you're doing multi-compartment modeling, you'll want to set full morphology. And if it's a pyramidal cell, you'll also want to make sure that the apical dendrite is intact. Now, finally, you can also do some putative assessment here, where you can say whether or not it's probably excitatory or inhibitory. If you select spiny, it's more than likely an excitatory cell type. If it's aspiny or sparsely spiny, it's more likely an inhibitory cell type. So with this search that I have here, this is an example of some results that you would get. And it looks like a putative excitatory neuron, a pyramidal cell. So you would just click on electrophysiology, which should take you to this page. And on the top left, it gives you some demographic data. So it's a layer 5 cell from middle temporal gyrus from a 34-year-old female with epilepsy. And then in the middle, it just gives some example E features extracted from those traces, as well as a steady state versus current plot, as well as a spike rate versus current plot. On the bottom, you see some examples of spike traces that you can look at automatically. So this is an example with a long square stimulus, but you can select different stimuli. They have a whole set of different stimuli that they use, for example, ramp stimuli or noise stimuli. And perhaps the most important part about this is you can download this data and it's openly accessible, and you can do your own analysis on the NWB file that you'll get from downloading. If you want the reconstruction as well, you can click on View Morphology, which should take you to this page, and you can just click on Download Morphology, which will give you the SWC file. And from there, you can do some multi-compartment modeling on that cell type. If you want to access population data, there are also ways which you can use Allen Brain Institute's toolkits that they have. I think it's called the Allen SDK module, to programmatically access the data from labeled cell types. Another option is if you have access to other published repositories that are online. For example, this is a repository of excitatory cell types from our collaborators over at UHN, Krembil Research Institute. This is from Dr. Valiente's lab, where you have, like I said, different excitatory cell types across different layers of human cortex. And you can do your own analyses on this electrophysiology data. It has a different protocol from the protocol used by the Allen Brain Institute to larger steps, so you have different options to choose from. So once you have that data, the next step would be to extract e-features or electrophysiology features. So like I said, BluePyOpt uses the eFEL module to assess the models that it generates. And that stands to reason that you should probably also use the eFEL toolbox when analyzing the experimental data, just to make sure you're not comparing apples to oranges. For example, spike rate, which is a pretty innocuous, straightforward measurement, is not always measured the same way. You can measure it from the start of the stimulus to the last spike, from the first spike to the last spike, and these small differences can introduce systematic errors if you're not calculating it the same way between the experiments and the model. So you really just want to make sure that you're using the same measurement. eFEL contains a bunch of different features. There's plenty of options to choose from. Some examples here: spike rate, adaptation index, time to first spike. We have action potential amplitude, after hyperpolarization depth, and action potential width. And there's, like I said, a whole bunch of them. You can check out their website and their documentation. It's pretty extensive. So just getting into the optimization setup now with BluePyOpt, the first thing you want to do when you have that data is to load the morphology and create section lists from it. So here's an example with a human layer 5 pyramidal neuron that I've renamed the file to pyramidal.swc. You'd load that with the NRN file morphology function, and you have the option of replacing the axon if you want. The default replace axon function in BluePyOpt preserves some aspects of the original axon initial segment, but there are ways to patch that axon replacement function if you want. There's also, so for the section lists, that's all down here. These are just pointers for when you want to insert channel mechanisms into your model. So for example, if I want to insert sodium channels into all compartments, I'd point it to all loc. And if it's a pyramidal cell, you'd also want to create an apical locator as well. So then finally, you can create mechanism lists, which has to do with the ion channels. This just points to where you're going to have ion channel mechanisms in the model. So you'd create a list, let's call it modmex, and then for each model mechanism that you instantiate, for example, passive properties to all locations, you would append that to that modmex list, which you'll then insert into another function later on. So here I'm showing examples of doing that with IH to all locations, sodium and potassium channels to the somatic locations, and then appending them to that list. Once you have all your model mechanisms, you can create parameters, which here you'll specify which parameters will also vary in the optimization. And most of these parameters are associated with the ion channel mechanisms that you already insert into your mechanism list. But you have some default parameters in Neuron, like temperature, which is a global parameter. You can set it, for example, here I'm setting it to 34 and I'm keeping it frozen so that it does not vary in the optimization. Here's an example of a section parameter that is also frozen and does not vary in the optimization, so the axial resistance. And here's an example with sodium channels where I do want it to vary in the optimization. I want the optimization to find optimal solutions of sodium conductance. So I'd set frozen to false, and I'd give it a boundary from, in this case, 0 to 0.7. And that's the lower and upper bounds through which it can vary in the optimization. And then you can do some other cool stuff. BluePyOpt has a lot of cool functionalities. So for example, with IH, I want that to scale with distance from soma, so increase the conductance with distance from soma. And I also want it to vary in the optimization. So same thing with sodium, I give it a boundary, I set frozen to false. And I also create a scalar, in this case with a mathematical formulation of a sigmoid, so that the IH conductance will increase sigmoidally with distance from soma. I'd append all those parameters to a list similar to what I did with modmex. Here I have modparams. And I'd insert all those into a cell model template. So these are all inputs. So modmex, modparams, as well as the morphology that you generated in that first slide I showed. So once you have your cell template, you can go on to create the recordings and stimulus protocols that your simulation will run. And it stands to reason that you'll want to have the same stimulus and recording protocols that you had in the experiments that you're trying to model. So with the same step amplitudes, the delays, durations, the patch location. Usually patch locations will be at the soma, but you can also do some cool stuff if you have access to dendritic data, if you want to model some features in the dendrites or even some features in the axon. You can also create different recordings. In this case, I'm showing an example of recording at step 1 at the soma associated with that stimulus. And then depending on how many recordings you have, stimuli you have, you can append them all to lists and input them into the sweep protocol function, which is the protocol that you will use when running your model. And then the next step would be to create a multi-objective fitness calculator. So for this, it gets a little bit more complicated, but essentially you want to group together features, and each group of features becomes an objective. So if I have a group of features that I want to capture the spike train features, I take something like the spike rate as well as the adaptation index, create eFel features with them, insert the mean and standard deviations as taken from the experimental data. So those are the targets for those features, and append them to a list, which I'll call features 01. 01 standing for objective 1. And then I'd insert that list of features into a max objective function. And max objective takes the maximum error between those two features, and that's given to assess the fitness of the model. There's different objective functions you could use, a weighted objective if you want. Again, BluePyOpt has lots of functionalities. Here I'm just showing one example of a type of objective that you can have. Then you can create other objectives as well, maybe to capture spike time features like the time to first spike, or even spike shape features like the action potential width, amplitude, and AHP depth. And so here I have a list of 3 different objectives by the end of it. And that's why we call it a multi-objective optimization, is because you're trying to capture multiple objectives in your optimization. You'd input that list of objectives into an objectives calculator, which is used to assess the fitness of the model. So that fitness calculator then gets inputted into the cell evaluator, along with the cell model template, a list of parameters that are not frozen, the recording protocols, as well as the simulator. And it's at this step where you'll also want to instantiate your the neuron simulator and input that into your cell evaluator. So once you have your cell evaluator, you can then run the optimization, which you'll start by inputting the cell evaluator here. You'll also, if you're running in parallel, want to input a map function, which is essentially just to parallelize the optimization such that on each generation, you're sending each model in the population to a different processor, so it can simulate as many as possible in parallel. For this, we use SciNet typically. In the past, we've also used Neuroscience Gateway, both great resources. And if you wanted some examples on how to create that map function, you can check out the BluePyOpt GitHub website. They have some examples there. And then finally, you can set some parameters that have to do with the algorithm itself, such as crossover probability, mutation probability. So going back to that original description I gave of evolutionary algorithms, as well as eta, which is the learning rate, which has to do with how quickly the optimizer converges onto a solution space. And then also things like the offspring size, which is just the number of models in each generation, the maximum number of generations you want to run it for, the evolutionary algorithm that you want to use, so that's selector name. In this case, I'm running it with the indicator-based evolutionary algorithm, as well as the seed value. And with that, you can just run the optimization using optimization.run, which should output the final population, the Hall of Fame, which is just the top 10 models that it generates, as well as a log and history of all of the models that it generated. So here's some quick examples of models that we've developed in the past. This is the human layer 2-3 fast spiking basket neuron. This is data obtained from the Allen Brains Institute and an example of a single cell optimization that I ran. So it's just optimized to one cell. Here's the morphology of that cell. Here's what the spiking looks like in the model. So it captures the spike rate actually quite well. You can barely see it here because the traces are overlapping pretty much completely. Yeah. And then finally, here's an example of optimizing models to population data sets, in this case from Dr. Valiente's lab. So here I'm optimizing layer 5 pyramidal cell models to young and old demographic data sets, where young is anything less than 50, old is anything older than 50. And here are some examples of what it looks like with the spike rates. It's within 1 standard deviation of the experimental data. So it captures the experimental data quite well for both the young and the old, as well as the seg amplitude. So you see the seg ratio is also within 1 standard deviation of the experimental data, and you see a bit of an increase in seg amplitude in the old model relative to the young. So in summary, the human neuron data sets are available and openly accessible, and there's sufficient human data available for the construction of full morphology neuron models. With that, with openly accessible software tools, this provides a comprehensive state-of-the-art method for automating the generation of neuron models. So we have a lot of options out there with all these resources that are available to us nowadays. So with that, I'd just like to thank CCNS for having me on, and I'd like to acknowledge the lab over at Krembil Center for Neuroinformatics, the University of Toronto, the Krembil Foundation for their generous donations, as well as the various resources that I presented in this presentation. Thank you.
Thank you, Alex. That was very illuminating. There are a couple of questions. What does the seed value do?
Right. So the seed value just sets the randomization. So if you set it to the same value, it should generate the same solution by the end of it, the optimization. Because there's some random aspects in the optimization for it to converge onto solutions, generating new models. So setting the seed value just allows you to replicate the same result twice.
Right, that's great. Another question, a lot of the setup of the model looks similar to the older neuron Hawk coding language. Which of these steps are specific to BluePy-opt or Python?
The setup of the model looks similar to the other Hawk coding.
Yeah.
Right, so I'm not sure which steps they're talking about. Certainly with the neuron simulator, you can, it's pretty interchangeable. So if you want to write something to do with the accessing the neuron hook, you can write it in your Python code, but like specifying that it's hook code. Most of the BluePy-opt code is pretty much Python based, I want to say. But there are some aspects within their source code that does have to do with hook if you want to do things like output it to a hook model template.
Yeah, that was a question from Scott. I think you answered it. Because
So.
yeah, I wasn't sure exactly, because there are a few aspects, right? But
yep, that was definitely me. No, that helps.
I can bring it to the slide if because I wasn't sure which part looks hawkish, you know.
Well, yeah, just a lot of the mechanisms for like putting the ion channels and whatnot in the various portions of the model, and you know the various labeled dendrites. It seemed to be at least similar, you know, coding language to the Hoke stuff, but all the optimization stuff is pure BluePy-opt.
I see. So like actually, it's good that you mentioned that, because a lot of the names that you have to give in those when you're calling those parameters have to be associated with the same suffix name that's given in the mod files. So yes, there is some consistency that you have to keep between what's stated in the neuron/mod file code.
Okay, so basically some of that, the lingering, mod hoc, whatever you want to call it, language from the model still has to be preserved in the way you're labeling things in order to implement loop-iout.
Right.
Okay. Awesome.
All right.
Stop
So
sharing.
yeah, this tool is really nice. It's free. It has been used by various labs, and it's pretty Python based. So most people that are happy with Python can use it very simply. I think we can maybe, because I don't see any other question, maybe finish here. We can have a little bit of a breather, a real breather. And 3:15, I think, is the next and last talk in this session. I'll just let maybe a minute for anyone who has maybe wants to squeeze a question, because we still have a few minutes for this session. Scott, if you want to also, I don't know, announce anything. I mean, you're muted, I think.
Good.
You're muted, Scott.
Oh man. Yeah, I'm falling victim to all the old tricks today. But yeah, so I'll just mention, as Itai said, the last talk of the session is from Steve Prescott at 3:15, so we'll have a little bit of a break. Following that is our first hour-long trainee talk session. We have 2 spotlighted talks to start, and then we will have 4 parallel sessions that you can pick and choose which topic you'd like to go to. And again, because all of the talks are available on replay, if you miss one session or you miss a talk that you want to see, you will be able to watch those quite literally minutes after they end. Crowdcast generates a replay. So for those people who are having conversation in the side about note taking and replay and things like that, that is one of the benefits to Crowdcast. So those replays should be readily available to you all. And if you have any questions about accessing them, feel free to ask me or any of the other organizers. So with that, I'll end this session, give everyone a solid 5-6 minutes to go take a break, and we'll start the next session right at 3:15. Thanks, everyone.
Take care. Bye.